In this figure, let's examine thick ascending limb of Henle transport. And the thick ascending limb of Henle and the loop of Henle itself is absolutely critical for water conservation by our kidneys. And let's look at how. So we have our loop of Henle shown here. And we're going to focus on what's referred to as a juxtamedullary nephron. This is a long loop nephron, the loop of which would project significantly into the medulla. So if I draw a line here, separating the cortex from the medulla, these juxtamedullary nephrons have these long loops of Henle that project down with the collecting ducts toward the papilla down here at the bottom. Now, we're going to examine some of the cells of the ascending limb here. We're going to blow them up and look at the ion transport that occurs there. But let's also look at the descending limb on this side and note that it is actively reabsorbing water. Now, we don't know why yet. We'll see that in just a moment. And that's really its key characteristic of the descending limb, is its water permeability. The ascending limb we'll see is transporting solute into the interstitium here, generating a driving force for water reabsorption, but it's not reabsorbing any water. And so they sort of do opposite things, the descending versus the ascending limb. So let's focus on that ascending limb over here because that's actually going to help us understand this descending limb water reabsorption. Now, the starting point for our tubular nephron function here is the sodium-potassium pump in the basolateral membrane. As always, that pump is reabsorbing sodium, pulling it out of the cell. That's going to be taken up into the peritubular capillaries here. These are known as vasa recta because they're in the medulla. And we're going to be pumping potassium into the cell. Now, we've already reabsorbed some sodium, so we've got part of our picture over here already, the sodium reabsorption. But secondarily, remember, we are generating a sodium gradient out to in that is going to be utilized as potential energy to drive the transport of other solutes. We're going to see that over here with a very specialized apical membrane transporter known as the NKCC2 cotransporter. Well, the NKCC is sort of telling us something here. Sodium, potassium, and 2 chloride ions are going to be co-transported into the cell from the tubular fluid, reabsorbed. This is driven by the sodium gradient potential energy. Sodium is moving down its gradient to move potassium and chloride up their gradients. Now, the chloride will be allowed to transit across the cell and escape at the basolateral membrane across a channel. Now we have added chloride to the interstitium, again, making that interstitial environment more concentrated. The potassium at the basolateral membrane is going around in circles here through a potassium leach channel. And that generates an inside negative relative to outside positive membrane potential, as we've seen many places before. At the apical membrane, the potassium is also sort of going around in circles, but in this case, it's leaking out. Again, that would make the inside negative relative to the outside, but it's making the tubular fluid electropositive, right? Our NKCC2 is electroneutral. There are 2 positives and 2 negative charges moving side by side, but the potassium backleak is making the tubular fluid electropositive. That's going to drive the reabsorption of other cations. It's going to repel them, especially across these paracellular pathways, driving their reabsorption as well. Now, if we come back over here to our smaller picture at the right, what we've done is we've transported solute out of the ascending limb, generating a gradient in the interstitium. And I could draw this anywhere. And it gets saltier the deeper we go into the medulla. It would also be over here, right, everywhere, over here next to our descending limb. Guess what? That is what is driving the osmotic water reabsorption from the descending limb of Henle. That increases the solute content concentration on the inside, fueling the transport of this NKCC2 cotransporter, right, driving that transport outward, generating the gradient that we see over here. And now, again, these loops of Henle generate a corticomedullary solute gradient circled here. And that is also going to favor water reabsorption from the collecting duct. This is hormonally controlled, but is the key function of the loop of Henle of these long loop nephrons. It allows for water conservation through osmotic reabsorption from the collecting duct, as well as the descending limb of Henle.